The question number 2 is about photosynthesis. The student must have the knowledge about the topic of photosynthesis. The question part 2.1 is an AO1 question. So, what are the two products of photosynthesis? The student must have the concept about photosynthesis and the chemical equation of photosynthesis. So, I have written this equation here. The plants take carbon dioxide from the air and they take water from the soil and they make glucose and they release oxygen in the air. These are my reactants and these are my products. So I have two products right now. One is glucose, another is oxygen. So I'm going to pick my products here. One is glucose and other is oxygen during the process of photosynthesis. Now this is an investigation. A student investigated the effect of light intensity. So this is the key information, the light intensity on the rate of photosynthesis. Figure number 4 shows the apparatus, LED light source, and the distance from the light source in centimeter. If we see in our apparatus, the pondweed plant is used in sodium hydrogen carbonate solution there. And I can see the bubbles form as a result of photosynthesis. This is the method used. Place pondweed 5 cm from the light source. Measure the rate of photosynthesis by counting the number of bubbles produced in every 30 seconds. Repeat the investigation with the pondweed at different distances from the light source. Means if you are taking at 5 cm, repeat at 10 cm, 15 cm, 20 cm, 25 cm. I keep going. Now we are going towards our question number 2, which is related to this investigation. And the question is, how could the student measure the rate of photosynthesis more accurately? It means the students need to measure how quick or how slow is the photosynthesis process going on and how accurately he can measure the rate of photosynthesis. It is an AO3 question, so this is investigation based question, and different options are given and the student needs to pick two boxes from here. And most of the students they try to make a mistake by picking up only one box. So there are different options are given, 5 options are given. So how to measure the rate of photosynthesis? First option is count the number of bubbles produced in 1 minute. Second option is measure the change in the mass of the pondweed in 30 seconds. Measure the volume of the gas produced in 30 seconds. Place the pondweed further from the light source. Use water instead of sodium hydrogen carbonate solution. Now the different things are coming there. So we have to measure the accuracy because photosynthesis is taken place. So we have to count the volume of the gas produced, which is oxygen. So this process is going on in sodium hydrogen carbonate solution. So you can see the bubbles there. The option which I'm going to pick here, that is count the number of the bubbles produced in 1 minute. Another option which is given to you, which you can pick for our answer is measure the volume of the gas produced in 30 seconds. because we can't measure the change in mass. There is no point to put the pond weed apparatus from the light source. If we are now using sodium hydrogen carbonate, the plant is unable to take carbon dioxide and photosynthesis will stop. So these are the two ways we can make the rate of photosynthesis more accurate. Our part number 3 is also an AO3 question in which the student is using LED light. The LED light source doesn't get hot. Why this is so important? Instead of the normal light, the student is using the LED light. This is an AO3 question and the whole question is related to the photosynthesis. So the different options are given. It is 1 mark. So I put different options and try to explain the reason behind it. First is the temperature affects the rate of photosynthesis. Now the question comes in the student mind, how the temperature affects the rate of photosynthesis. The answer is very simple. If the temperature is increased, the rate of photosynthesis will increase and the number of the bubbles will increase and it can affect the rate of photosynthesis. Second thing is, at very high temperature, the enzymes will be denatured and the rate of photosynthesis will be affected. Or the student can use another option there. If the temperature is too low, the enzymes will not work even at a very low temperature properly. So the rate of photosynthesis will be affected again. So the best option is the temperature will affect the rate of photosynthesis. Question 2.4, which is an AO2 question, now the students need to apply the knowledge. Calculate the number of the bubbles produced in 2 minutes when the light source was 10 cm from the pond weed. It means that students need to use the previous information which is given. The previous information is a table is given. The distance of the light source in centimeter is given here. I highlighted that is 10 centimeter according to the demand of the question, and the number of the bubbles produced in 30 seconds is mentioned here in front of them. Now the question is coming for 2 minutes and the distance is 10 centimeter. Therefore I highlighted 10 centimeter distance and I have to take it 2 minutes there. So I can see that in each 30 seconds there are 13 bubbles are coming. So 2 minutes mean that is 120 seconds. So it is very easy for us to find it. The 13 bubbles are formed in 30 seconds. In 120 seconds, which is actually 2 minutes, it means 4 times the bubbles are coming there. The reason behind it is that 120 means 30 second, 30 second, 30 second and 30 second there. So 4 times 30 is coming there. That is going to make 120 seconds. So the number of bubbles will also get 4 times. That is 52 bubbles are formed in 2 minutes. The next question is 2.5, which is also an AO2 question. Plot the data from the table number 1 on the figure number 5. So graph paper is given to you here, figure number 5, and you need to plot the data from here, which is from the previous information which is given here. Table number 1, the distance is given and the number of bubbles in every 30 seconds. You need to plot the graph between them. You need to draw a line of best fit or after plotting all of these points on the graph. So I have already plotted this thing for the students. 5 centimeter, there are 40 bubbles. At 10 centimeter, there are about 13 bubbles. At 15 centimeter, there are 5 bubbles. At 20 centimeter, there are 2 bubbles. At 25 centimeter, there is 1 bubble. And when the plot is about 30 centimeter away from the light source, there is no bubble formed there. Then I draw the line of best fit, which is a curved line. A lot of the students, they make the mistake, they try to draw the straight line here to here, which is wrong. You need to see the trend going on. The trend is the line is going down, passing through the points like this way, and is going to touch 0 here. And if you look here, as the distance is increasing, the number of the bubbles formed is going down. The part 2.6 is, which is AO3 question, correlated to this investigation. Give one conclusion that can be made from this result. So the result is given here, which is very clear result. As the light source is moving away, or as the plot is moving away, the number of the bubbles produced during the photosynthesis is reducing. So that is very simple trend. So what we can write here, that we're going to see the model answer. As the distance of the light source is increasing from the pond weed, the rate of photosynthesis is decreasing because the light intensity is decreasing. Or you can say the other way around. If the distance of the pondweed is decreasing from the source, the rate of photosynthesis is increasing and more bubbles are formed. As the distance of the light source is increasing from the pondweed, the rate of photosynthesis is decreasing. As the distance is increasing, the rate of photosynthesis is decreasing because the light intensity is decreasing there. Or you can go the other way around. As the distance is decreasing, the rate of photosynthesis is increasing. The question number 3 is again an investigation, and it is regarding the concept of the osmosis and diffusion process. A student investigated the effect of different concentration of sugar solution on a piece of potato. If you look here, this is tube number A, tube number B, and tube number C is given. The method the student has used: cut 3 pieces of potato to the same length, dry each piece on a paper towel, weigh each piece, place each piece in a different concentration of the sugar solution, leave all 3 pieces for 2 hours, remove the 3 pieces of potato from the solutions, dry each piece on a paper towel, measure the length and the mass of each piece of the potato. The figure number 6 shows how the investigation was set up. So if I'm going to look here, I highlighted some of the important area for my students. Tube number A, tube number B, and tube number C. The potato cylinders or pieces are already there, same size, but the important thing is we have to look into the concentration. it is 0 mole per dm cube. 0 molar solution is present here. 0.4 mole per dm cube solution is present there, and 0.8 mole dm cube solution is present there. So it means there is no sugar, only the water is present. Now the first question is coming, which is an AO3 question, 3.1. Why did the student dry each piece of potato before weighing it? The student must have the concept of osmosis. The reason behind it is, after the experiment, the student need to dry all of these one, which is given here, point number 7. Dry each piece on a paper towel. If these potato pieces or cylinders are wet, so they will increase the mass of the potato. The student has to do this thing because the amount of the water on the potato will increase the mass of the potato. To get the correct mass of the potato, the student needs to use point number 7. Let's see the model answer. The model answer is, which the student can write for the exam, and it is 1 mark question. The amount of the water on the potato would increase the mass of the potato there. To get the correct mass of the potato, the students need to use the point number 7. The second part of the same question 3 is an AO2 question. The students need to apply the knowledge. What 2 changes would you expect in the potato in tube A after 2 hours? Again, the student must have the knowledge of osmosis. So the student has to observe what will happen to the potato cube or potato cylinder in tube A. If you go back there in the figure, which I have already highlighted, the water concentration outside here in the tube is very, very high. So these arrows are showing the water is moving inside here. From the water here, it is going to move inside the cube. And how it is moving? It is moving as the result of osmosis. When the water will move inside, so the weight is going to increase, the length is going to increase. Let's see the different options which is given. We need to pick 2 boxes from the options. Now we're coming back to our options. The first option is the potato piece is going to break into pieces. It is not going to break into pieces. Decreases in the hardness of the potato. There is no decrease in the hardness of the potato. As the result of the osmosis, water is moving inside, the size will not decrease. As water moves inside, the mass of the potato will increase. That is the correct option. As the water will move inside, the length of the potato cube will increase. So these are 2 correct answers for 3.2. Part 3.3 is to complete the sentences which are given below. Water moves into and out of the cell by the process. So it is an AO1 question. So that is osmosis. A lot of the students try to write the word diffusion, but the examiner will not going to give the mark, because for water you have to write the specific word, that is osmosis. This is regarding the tube A, so it means the water is moving into the potato cells. Therefore, the size and the mass of the potato cylinder will increase. The solution outside the potato in the tube A is at low concentration than the solution inside the potato cell. And if we go back here, the figure number 6, and it is very clear that water is trying to go inside as the result of osmosis. And what will happen? Why the water is moving inside? Because outside the concentration is very low. This is 0 mole per dm cube. So there is no sugar present there. As the result, water is moving inside. Our next question is 3.4, and it is an AO3 question. The student have to give a correct reason behind it. Again, this is about the concept of osmosis. The potato in the tube B didn't change. The size and the mass is not changing. Give one conclusion that can be made from this observation. If the size is not changing, if the mass is not changing, it means the osmosis process is not taking place. And what is the reason behind it? The reason is the concentration of the sugar solution inside the potato and outside the potato is same. Therefore, the diffusion process will not take place. So the model answer we can give there, the concentration of the sugar solution inside and outside the cell, mean in the tube, is same. This means that 0.4 mole per dm cube is present inside and outside. The rate of osmosis will stop. Therefore, the size of the potato in tube B will not change. Our next part, 3.5, which is an AO1 question. The student must have the basic knowledge about this topic. The figure number 7 shows the root of a germinating seed. So this is the diagram which is given here. A root is given here. Along with the root, okay, germinating seed, this is the root. Along with the root, you can see that a lot of the root hairs are present. The question is, describe 2 ways the root is adopted to absorb water efficiently. Here, the student needs to use the concept of osmosis and diffusion and the specialization of a cell. So root here is a specialized cell, so it has specific function. So what are the things? Describe 2 ways the root is adopted to absorb the water efficiently. If I look into the diagram, the root here has spread a lot in the soil and they're quite thin. The cell membrane is very thin there, and they have covered a lot of area in the soil. Therefore, the chances are they can absorb more water for the seed. So the model answer we can give there, the root has a lot of thin root hairs. The root hair can spread in the large surface area in the soil. These root hairs have thin cell membrane. Therefore, it is very easy for the water and the minerals to move into the root hair from the soil. The root has a lot of thin root hairs. The root hairs can spread in the large surface area, which we can see from the diagram. The third option which the students can use, the root hairs have thin cell membrane to absorb water and the minerals from the soil easily.